Anirban. So at first, I would like to thank the organizers of the ICMRBS committee. So to give me the opportunity to present my work, which I did sometimes in last few years. So my topic is the structural studies of the nucleic acid ligand interaction by NMR. So let's start with our system. So let's start with about myself. So what I did. So I did my PhD in Kolkata from India, Bose Institute, and where I worked on the different types of the peptides, antimicrobial peptide and self-assembling peptide, and some parts of the protein misfolding by NMR and other relevant biophysical techniques. Then I moved for a postdoctoral work in Bordeaux, France, but to work on a completely different systems on nucleic acid biophysics, but not on the NMR, on the other technique by mass spectrometry, by native mass spectrometry, as well as some applications of the ion mobility spectrometry to understand their shape and structure and their polymorphism. Then last year I moved to my second postdoc in Czech Republic, Brno, in Central European Institute of Technology. Here I am developing the in-cell NMR methods to study nucleic acids, as well as development of the 19A probes to study conformation of the nucleic acids. So let's start with the topic of my talk. So as we all know that nucleic acids, they are very important in life because they contains all the genetic information and they carry over from generation to generation. So during the last 20 years, there has been a considerable interest in studying the non-canonical nucleic acid apart from the regular Watson-Crick duplex model. So there are several types of nucleic acids, for example, like G-quadruplex, I-motifs, some types of Z-DNA, and different types of triplex, antiparallel conformation. So today I will talk about the G-quadruplex. So let's see why it is important in life. So because these type of sequences are found in different types of the human and other genomic sequences like oncogene, promoter sequences, and telomeric sequences. So they have definitely some biological roles. They are involved in some cellular process like transcription, translation. So that's why they can be treated as an attractive drug target too. But apart from the biology, they are a good supramolecular system because they are polymorphic by nature. So one can tune their property by adding cations by changing pH or adding solvent. And currently nowadays, people are using this type of system for the nanotechnology application. For example, for the creation of the DNA origami type of superstructure, as well as the G-wire nanomachine type of conformation. So they are important in all parts of the chemistry and biology. So let's see the structural part. So what is there in the structure? So as we know that in regular Watson-Crick DNA duplex, there is a Watson-Crick base pair, which is actually stabilize the structure. But in G-quadruplexes, we have a different kind of base pairs. These base pairs called the Hook-Steen base pair. And these Hook-Steen base pair found between the guanines. 4 guanines are involved in the 8 Hook-Steen base pairing, and they are and the adjacent guanine strands, they are stabilized by further coordination with the cation. So we can have different types of conformation. For example, the parallel conformation where both the strands are in a same orientation. We can have hybrid where 3 strands in one orientation and one strand in a different direction. As well as we have antiparallel conformation where there is 2 strands in one direction and 2 strands in other direction, or by this type of basket conformation where one is one and another in a down direction. So we can have different types of the 4 different types of the conformation of this type of system. And for all this system, if we can see the whole structure of a particular quadruplex sequence looks like this. You can see that there are centrally three quartets which are stabilized by involvement of the hydrogen bonding as well as the 2 cations in between the quartets. So this is basically a hybrid quadruplex structure. So in our system, we work with the telomeric G-quadruplexes, which forms from the repetitive sequence from the TTA-GGG repetitive sequence. So it can adopt at least 6 different G-quadruplex conformations till now, which has been solved by the X-ray crystallography and NMR structure. So some of the conformations are below. If you see that they are there is a variety of conformations, but the central unit is the same, but the difference is only present in the overhangs and also the loops region, which gives rise to a conformational polymorphism, but also the presence of the cation. Some structures are solved in presence of the cation, also different cation, like because for example, most of the structures are solved in presence of the potassium, but there are few structures which solve in presence of the sodium ions, and some structures solved in presence of the molecular crowding agent like PEG also. So it's extremely polymorphic in nature, human telomeric quadruplex. And there has been some structures solved during the last 10 years. That some ligand bind conformation is also solved to date. And in most of the cases, we can see that the ligand is actually binding on the 5' part of the quadruplex, which is marked by this circle, and stabilize the quadruplex by means of the external stacking. But there are some caveats. For example, we have several antiparallel quadruplex structure, which shown to be very important in biology and chemistry and can be termed as one of the important parts of the folding energy landscape. And recent study show that they can be observed inside the cell, inside the cellular milieu also. But there has not been any high resolution conformation in presence of the antiparallel quadruplex in presence of some small molecule ligands. So during our project, we collaborated with the Professor Mary Paul from Institut Curie, Paris. She gave us this beautiful small molecule. This molecule, we will call it phendicity from now onwards. So if you see this molecule, so this molecule is a planar molecule, and with having two large aromatic moieties. One is the phenantholene moiety and stabilized by these two arms. And this molecule is known to bind on the N stacking mode on the 5' quartet. And this is a good ligand because it has some selectivity over the quadruplex versus the non-quadruplex or duplex DNA. And also it is involved in the helicase mediated G4 unwinding also. So our previous study from our group shows that it can induce conformational transition from hybrid to antiparallel G-quadruplex. But we don't know how it can induce and what is the final conformation. So I will just briefly discuss a bit about the mass spectrometry, because I guess most of the people here are experts on the NMR. Because in mass spectrometry is a beautiful technique to study the conformation of the biomolecules. So we use a volatile buffer supplemented with a salt. And we can count the number of cations, and from the number of cations, we can count the number of quartets, because I mentioned that the quartets are stabilized by the coordination with the cation. So if we can count one cation, we can see that if we can count the two quartets, we can see there is two cations bound in the 3 quartet. So it can be a 3-quartet quadruplex structure. And if we see one cation, we can see it's a 2-quartet structure. But as well as we can see some unfolded structure also. So we can observe simultaneously all the species present in the solution by mass spectrometry. But also we can observe polymorphic quadruplexes also, which can give more than one conformation, which is difficult to observe and solve by NMR and other techniques. But we can see from the mass spectrometry that there is at least 2 conformations. One is a 2-quartet and one is a 3-quartet conformation. So we start with the titration in mass spectrometry by using the ligand I mentioned, phendicity, and also a hybrid 3-quartet human telomeric G-quadruplex, which is a hybrid structure. So if we did the same titration in mass spectrometry and in NMR. So we observe the initial structure is a 3-quartet conformation stabilized by the 2 cations, and when we sequentially add the ligand, we observe a new peak appears. This new peak contains only 1 cation. So which means the structure is defined. There is 2 quartet and 1 cation and 1 ligand. And upon prolonged addition of the ligand, we observe that more than 1 ligand bind to the quadruplex. And also in the CD, we observe that there is a very different change in the conformation. One is from the hybrid initial structure to the antiparallel structure. And we calculate the binding affinity also from this mass spectra, and we can see the first binding affinity, the binding of the first ligand to the quadruplex is very strong in the nanomolar range, but the second binding affinity is relatively weak in the micromolar range. Then we perform the 1H NMR titration to see that how it looks in the spectra. So the initial structure is a hybrid quadruplex structure. When we add sequentially add the ligand, we observe that slowly a new peak appear in the spectra. So there is a two sets of peaks. One state is the free state and one state is the bound state. And at 1:1 concentration, we observe a completely new set of peaks, which comes from the bound quadruplex as well as from the ligand. And upon further addition of the ligand, we observe a peak broadening due to conformational exchange between the several ligand-bound substrates. So we know the initial structure, that's the hybrid one, but we don't know the final structure and how it interconverts to the initial structure to the final structure. So we collaborated with the Slovenian NMR Center with the Professor Janez Plevek and Marko Tobolski, his postdoc, to study the nucleic acid, to study this system by high resolution to the NMR and also synthesizing some label compound. So we synthesize partially guanine level at the C13 and C15 for our sequence. So as I mentioned, since there is 12 guanines, we synthesize 12 label guanine compound, each of which is labeled with the C13 and N50. And we did the 15N and 13C filtered HSQC experiment. And from this HSQC, we assign all the protons in the immuno region, which is actually the fingerprint region of the quadruplex, as well as the aromatic region. And also we find the assignment of the ligand from the other to the NMR experiment. And we found some very critical observations. So we observe 8 sharp peaks, which is easily can be accounted for the 2 quartets, because in one quartet we have 4 guanines are there, so 4 peaks, and for 8 sharp peaks corresponds to the 2 quartets. That's okay. But we don't observe any peaks for the G3 and G11. On the other hand, we observe some broad peaks for the G15 and G23. It can be due to the dynamic base pairing in this region, which is very dynamic in the time scale of an NMR. That's why we observe very broad peaks in the immunoproton spectrum. So then we wanted to know that how these guanines are arranged in the ligand bound state. So if you see my cartoon, you can see that if the guanines are arranged in a syn-anti fashion, so that means there will be a strong peak of this inter-residue noisy peak because of the distance, and there will be, for the anti, there will be a weak peak in the H8 to H1 prime, the anomeric region. So we can have a look on the noisy spectra of the anomeric region, and from this region we can assign the topologies, the syn and anti topologies of the guanine. So we concluded that out of 12 guanines, the 6 guanines they are in the syn orientation, and the other 6 guanine they are in the anti orientation. So the quartets are arranged like this, like the 2 syn, then 1 anti, like 1 anti syn, like this type of arrangement. So then we wanted to know that how guanines are involved in the quartet formation. So which of the guanines are there in a particular quartet? So we again take help of the noisy spectra. So we take help of the immunoproton to the aromatic NOE, because in the one quartet there is a noisy pattern between the immuno of one guanine to the receiver hydrogen bond guanine of the H8 of the receiver hydrogen bonding guanine. So we assigned the spectra, and we found that in one of the quartets, the hydrogen bonding is anti-clockwise, but in other quartet, the hydrogen bonding is clockwise. And as I mentioned, for the third quartet, we did not observe all 4 peaks. We observed just only 2 peaks. So then we try to find the intramolecular NOEs. These are very important, as we all know, for the determination of the structures of the complex. So we found several around 73 intramolecular NOEs between the quadruplex and the ligand. And this NOEs is almost 2 times of the NOE that is observed in the few years before for the simic G-quadruplex ligand complex. So if we see it from a schematically, we observe that the ligand is actually intercalated in between the 2 quartets, and thereby giving the giving all the NOE contacts with the mainly the guanine residues of the quartet. So then we finally came with the structure of the complex, and we use more than 400 restraints and 73 ligand to DNA NOEs. So let's have a look on the ensemble of the structure. You can see the ligand is beautiful. The ligand is perfectly intercalated in between the two quartets, and thereby stabilize the conformation of the intercalated complex. And let's see that how the other stabilizing factors. So first, the stacking factor, the stacking of the ligand in the two quartets, this G16, G22, G4, G10, as well as the top 5' quartet, G7, G21, G5, G9, which is a dynamic quartet. We observe only 2 hydrogen bonds for the quartets. And also seen anti-stacking, which gives rise to some stabilization. But also it's not the only stabilization factor. There is some, if you see that one of the quinoleum rings of the Fendicity is slightly tilted with respect to the other ring, and just to give maximum pi stacking overlap. So this movement is called the correlated sig movement. And also there is some types of stabilization in the loops also, because in the loops there is a TT base pairing in the two TTA loops, two lateral TTA loops, and this is further stabilized by the continuous stacking with the adenine 8 and adenine 20. And the capping residue, the T13, A2, and A14, they also form a triad which further stabilize the 5' quartet and the ligand from the exchange with the solvent. So if we see our structure with the already published structure, so this is the first structure published around back in 10 years before. So in this structure, we can see the ligand is only stacks with the top 5' quartet, G quartet stack. Then this is our structure. We can see that ligand not only stacks, it actually intercalated in the G-quadruplex structure, and thereby it further stabilizes it. And after our structure, there are some new structures also. They take a quadruplex and also a long duplex stem, and they also have the same observation that ligand actually binds to the quadruplex duplex junction to facilitate maximum amount of the aromatic pi stacking. So we know the structure, but we don't know that how it converts the structure. So we want to know it. So there are some previous studies from the SMFRED, the single molecular spectroscopy, as well as some the replica exchange molecular dynamic structure, which showed that this quadruplex folding is extremely rugged. The folding energy landscape is rugged. So that means there are several intermediates and several minima which are actually stabilized by the kinetic partitioning mechanism. So, and there exists some type of intermediates also. They show that the G-triplex and this hairpin can be the intermediates of the quadruplex folding. So we wanted to see that whether our ligand can bind to the triplex and the G-triplex and the G-hairpin system also. So some recent NMR structure shows that for our system, this type of the triplex and this type of the hairpin system exists. And surprisingly, we observe that the ligand is actually binding to the G-hairpin system, and thereby convert to the G-hairpin system to the G4 system. Because if you see the similarity between this G-hairpin system and our complex, they are very similar in terms of the arrangement of the residues. So this ligand, because this has a large aromatic pi cloud, so this acts as a templating group for the assembly of the guanines to form the quartet. And then we also wanted to see that whether it can bind to the other misfolded forms. Some mass spectrometry study shows that the formation of the hybrid quadruplex goes via the antiparallel two quartet complex. Because if you see, vary the vary the salt concentration, we can see that the different salt concentration is actually stabilized the 1K form, which is actually the antiparallel conformation, which we observe from the deconvolution of the mass spectrometry and the CD data, and we found that the at different salt concentration, the ligand is actually bound to the 1K antiparallel conformation, and it stabilized the 1K antiparallel conformation as seen from the CD and the mass spectrometry data. And also it is reflected in the 1H NMR data. Also, you can see that at very lower submillimolar concentration of the salt, when there is partially folded and misfolded species, the ligand is actually binds and gives to the final topology of the antiparallel ligand bound complex. So we observe that, so we have one conclusion that this phenticity ligand can also bind to the intermediates, the misfolded species, which is the 2-quartet misfolded species. So finally, we came with the conclusion that our ligand is the first complex of the high resolution NMR derived structure of the antiparallel 3-quartet antiparallel G-quadruplex ligand complex. And what we observed from the mass spectrometry that the 1 potassium is ejected, and we actually see from the structure from the NMR also because of the intercalation of the ligand. But this is not only, we observe that this ligand can recognize the misfolded and the pre-folded species, because this is the important. Earlier many of the drug design access, they only consider the final folded quadruplexes, but we have to consider the misfolded and that pre-folded species. So it does not need to have a full folded quadruplex to bind to a ligand molecule. So it can recognize the misfolded species and redirect their conformation to the final complex. So this goes, so we conclude that rather than completely unfolding the quadruplex and then refolding, we conclude that it goes via a conformation selection model. It forms to a minor form, and then by the principle of the chemical equilibrium, it stabilizes the conformation, and whole system is moved to the particular conformation by the last atelier principle. So it goes via the conformation selection model. So finally, there is something left to do also, because if you see in the living cell, the polymorphism is much more, because there exists several folded species, several misfolded species also, and so we now I am planning to see, I am trying to see by in-cell NMR that some ligand can preferentially bind to the some parts of the quadruplex or some other parts of the quadruplex, which is still yet to be explored. So finally, I want to thank the previous lab in France, as well as my collaborators from Slovenian NMR Center and Institute Curie, and finally the funding agency from European Commission for giving me access to the facilities and etc. So thank you all for listening, and I am happy for any question.